Здравствуйте, дорогие коллеги. Спасибо, что присоединились к нам. И мы очень рады такому... Кто-то сказал, что там, по-моему, 200 участников у нас. Это замечательно для нашей школы. Школа наша сразу... Это уже третий год мы собираемся. В этом году замечательный букет лекторов подобрали мои коллеги, то и я тоже помогал. Надеюсь, вам будет интересно. Школа наша двуязычная, у нас много иностранных коллег, поэтому я перехожу на английский. И заодно для вас будет хороший урок английского языка, надеюсь. Вот, послушай. So, dear colleagues, thank you very much for joining our school. We are very happy to see you here, and I hope it will be interesting for you. And I know many of my friends, and I'm very happy to see them, will listen to these school lectures, and it's very interesting time to be, you know, in COVID time, it happened to be like we are separated, but at the same time united. Now we can invite the best colleagues from United States, from Belgium, from Italy, from Canada, from many other countries, and we can be together enjoying science. So at this moment, I'm stopping and moving to my lecture. Let me say where it is, demonstration of the screen. Yeah, do you see it? Женя?
Not yet.
Okay, now?
Yeah, now we can see it.
Yeah, now you can see it. It's good. So today I would like to describe the major topic of our lab, what we are working, trace amines and their receptors. And some interesting thing happened the last year, and I would like, it's not traditional talk, I put several interesting new things, what we have done recently. And okay. So trace amines, small lipophilic substances, very close to dopamine, serotonin, and norepinephrine. The most known of them are beta-phenylethylamine, tyramine, tryptamine, and ectopamine. They're known actually for about 130 years. Just imagine, dopamine and serotonin were discovered 30, 40 years before. And they termed as false neurotransmitters because it's believed that they working by modulating the function of well-established classical monoamines, dopamine, serotonin, norepinephrine. And it's believed that they do not have their own neurotransmitter function. But as I said, they were discovered before, and the beginning of pharmacology, classical pharmacology, happened actually with tyramine and beta-phenylethylamine, because the fathers of modern pharmacology were working with them when they were introduced to rabbits, or at the moment that was rabbits, they were increasing pressure, blood pressure, and that's how pharmacology started actually. They present in food and produced by human microbiota. I'll stop on that later. Below, you can see here below how close they are to classical monoamines. The importance of trace amines in psychopharmacology is difficult to overestimate. Just these facts. The beta-phenylethylamine, the best known trace amine, is the name of that, become part of the name of dopamine and amphetamine. Dopamine is just 3,4-dihydroxyphenylethylamine, so it's abbreviated dopamine. Amphetamine is just one alpha-methyl group, differentiate from beta-phenylethylamine, alpha-methyl-phenylethylamine. Only because of this alpha-methyl group, amphetamine is not metabolized by monoamine oxidase. So it's just, one can consider amphetamine as unmetabolizable beta-phenylethylamine, or beta-phenylethylamine as endogenous amphetamine with short lifetime. So it's being very quickly metabolized, quickly metabolizable amphetamine. Paul Janssen, who discovered haloperidol, for example, believed in the beta-phenylethylamine theory of schizophrenia. He thought that abnormal metabolism of beta-phenylethylamine, once accumulates, working like amphetamine-like way, increased dopamine. That's the reason of schizophrenia, just for example. Trace amines are 300-400 times, have a low concentration in the brain, about 300-400 times lower than dopamine, for example. but the rate of synthesis is comparable. It just cannot be stored in vesicles, and it has not, the release is not sensitive to depolarization. That is why they're not considered as a classical neurotransmitters, as a false. Two major enzymes involved in a lifetime of trace amines. They all produced from amino acids by decarboxylases. So L-aromatic acid decarboxylase is a major enzyme in the synthesis, and they metabolize like monoamine oxidase, the same way like dopamine, serotonin, norepinephrine. In fact, beta-phenylethylamine, the best known trace amine, was discovered in 1876 by Marceli Nensky. Marceli Nensky, famous Polish biochemist, was working in Bern in Switzerland at the moment, and he was studying decomposition of proteins, and he found beta-phenylethylamine as a product of decarboxylation of amino acid phenylalanine from white egg, from egg white. Interestingly enough, later he was invited by Ivan Pavlov to Institute of Experimental Medicine when it was created. And the right side, you can see Ivan Pavlov in white sitting in this hall with a lot of gentlemen. But one of them is Marceli Nensky, but I cannot recognize who is who here because all of them are bearded. They look all the same. Trace amines can be produced also by bacterial decarboxylases. So a lot of bacteria have their own decarboxylases, and they chopping off amino acids all the time. And so the same, that's why the concentration of trace amines is high in products where involving bacterial fermentation, such as cheese, wine, cured meat. But they also present in seafood. For example, octopamine is it was named after octopus, where it was discovered. Chocolate, for example, this chocolate there is high concentration of beta-phenylethylamine in the chocolate. Just as an example, soybeans, soy products, beer, for example. Look what beer contains, how many trace amines. Different trace amines. And it is believed that they working like, as I mentioned, like amphetamine-like ways. So they were like indirectly through transporters pumping out dopamine, norepinephrine, serotonin, and that's believed to be their major action for a long time. They were considered. In fact, the effect of tyramine and beta-phenylethylamine on norepinephrine became the basis of cheese effect in the periphery. So it was now it's almost very rarely used, but it's still prescribed sometimes. Monoamine oxidase inhibitors were used as antidepressants, and there were many observations that monoamine oxidase inhibitors with food, when they were consumed, were taken with food containing high concentration of tyramine, they create so-called cheese effect. So tyramine is pumping out norepinephrine in the periphery, in the blood, and you in the periphery, and in blood norepinephrine goes up, and it's vasoconstriction and acute hypertension, and sometimes people even died from that. And it was a warning, actually, when people were using monoamine oxidase inhibitors. They were warned not to eat cheese, beer, fermented food, smoked processed meat, and so on. So that was classical. And as I mentioned, that bacteria produce trace amines, so that's well-known fact. And now microbiota is coming into attention in many aspects. And microbiota gut-brain interface, microbiota interface. And what the role of trace amines endogenously produced by microbiota, it's not known. I'm expecting big developments here soon. So to sum up, so it can be endogenously produced by our own decarboxylases from amino acids. It can be taken from food, produced by gut microbiota. But think about that. It can be pathologically produced also by bacteria during infections. It can be in our skin. Microbiota is on our skin too. So it's another interesting aspect. to think about that. And that's all was known about trace amines until 2001, when a company which was working on deorphanization of G protein coupled receptors. Its deorphanization is finding ligands for unknown receptors. Okay, there are a number of orphan GPCRs which are not characterized yet, and there are several hundreds. And they were looking and trying to map it with ligands, particular ligands, and they were synaptic, and they synaptic preparation. And they were trying to find new serotonin receptors, in fact. And they discovered new family of receptors, and it's a different number in different species. In humans, 9 genes, but 6 of them are functional. And the maximum number of genes is in zebrafish 122, and 0 in dolphins. They all share a common motif. They all located in a single chromosome. In different species, different chromosomes, they go one after another like olfactory receptors. We were, at the moment I was at Duke working with Mark Caron on dopamine system, and we were invited to write a small commentary, and we wrote commentary which we named "Following the Trace of Elusive Amines." And since that time, for 20 years, I'm following the trace of elusive amines. And the longer I go by this path, the more exciting it becomes. G protein coupled receptors, or another way to say serine transmembrane containing receptors, 7-TMR, as Bob Lefkowitz would like to name. Bob Lefkowitz received Nobel Prize. He was at Duke University, and Mark Caron was his first postdoc, and we were working together. It's pretty much the same laboratory. Bob Lefkowitz received Nobel Prize for that. Mark Caron, unfortunately, not. So what Bob and Mark did, they discovered the first receptor, beta-2 adrenoreceptor, through which norepinephrine and epinephrine works. And then they proposed that there are many of them, and they were right. Now, right now, 826 GPCRs are known. 350 of them all over our bodies, somatic GPCRs, 150 orphan GPCRs. So 30–40% of current pharmacology based on targeting GPCRs. That's why it's not surprising that Bob Lefkowitz received Nobel Prize. Bob and Mark characterized for all these years, they were working together. It's the same place where they're working the same floors, just 2 neighbor laboratories, and all meetings are together. So they characterize how the receptors activated, how they produce signal, how they desensitized, what is known about arrestin in the induced signaling. That's another topic. So they did all together, and I'm very proud to say that for 12 years I was working with them, and we did a lot of interesting things together. Now, how they look now? Look at that. That's a family of GPCRs, and trace amine-associated receptors. They circled here. You can see a circle. They surrounded. Look, by which receptors? Beta-2 are other B2. That's beta-2 adrenoreceptor, adrenergic receptors, histaminergic receptors, serotonin, dopamine receptors. So they just in the middle of all these receptors, which become major targets for pharmacology for many years. Hundreds of drugs were developed surrounding on these receptors, and now this last part of biogenic receptors, which remain uncharacterized. So it's an untapped source for pharmacology. So as I mentioned, humans have 9 receptors. They have different old names at the beginning, T1, GPR58, 57, so on. Now they name just TAR1, TAR2, TAR3, just by order of appearance on chromosome. And as I mentioned, humans have 9 genes, but 3 of them are not functional. They're called pseudogenes. So humans have 6 functional genes: TAR1, TAR2, TAR5, TAR6, TAR8, and TAR9. It's believed that they are the closest gene for them. Like the predecessor was TAR5HT4 serotonin receptor. Just show how close they are to classical monoamines. And what receptors were discovered and knowledge started to accumulate about their activity on TARs in one or another function. Very interesting picture emerged. So in fact, it looks like that every amino acids, if you amino acid, you chop off, you take out acid part, you're left with amine. That's our trace amines. So for example, from arginine, you get putrescine. From phenylalanine, beta-phenylethylamine. From tryptamine, tryptophan, tryptamine. From tyramine, from tyrosine, tyramine. From valine is butylamine. And look, many of them already were found to be activating one or another TAR in different species. As I mentioned, there are many different species, many receptors in many different species. So arginine, agmatine, even histamine originating from histidine was shown to be activating one or another TAR, and so on. So pretty much we consider it as our table of Mendeleev. So we're predicting now that 20 amino acids will have their own decarboxylated amine, and these are new neurotransmitters, new biogenic amines having biological function. So not only amino acids, there are several other compounds originating from choline, for example, even metabolite of dopamine, tyramine, and so on. Several other compounds also can activate TARs. So we believe that the class of trace amines will be increasing in the future. So the best known TAR1 actually was characterized the best, and just making the long story short here. So we started, our colleagues studied from Roche, and it's clear now that TAR1 is a potential target to treat schizophrenia. So TAR1 agonists were shown to have potential in preclinical studies, potential antipsychotic-like activity, potential antidepressant-like activity, potential precognitive-like, and influence wakefulness. So TAR1 can, targeting TAR1 by agonists can help in various addictions like amphetamine, methamphetamine, nicotine, alcohol, even food intake in binge eating protocol can be affected by TAR1 agonists. TAR1 can be involved in metabolic disorders like diabetes, obesity. It's expressed highly in pancreas and stomach, and also in brain areas important for food intake. So also interesting application. There are a number of observations that TAR1 can be involved in the immune system. It's expressed in leukocytes, all TARs actually, not only TAR1. But the real breakthrough in TAR field happened last year, even before a little bit, when the paper published in New England Journal of Medicine, and which showed high efficacy of TAR1 agonists in schizophrenia. And right now, TAR1 agonists are tried in clinical trials. For Roche, Hoffmann La Roche has phase 2 trial with drug called ralmitarone, and Sunovion phase 3, ulatorone. And ulatorone by company Sunovion, it actually has not only TAR1 action, also activates several serotonin receptors, but TAR1 is a major drug target. It's like 10-fold higher activity on TAR1 than on 5-HT1A, for example. And they have several clinical trials right now, 17, I think, and here only 2 mentioned. And the data were so encouraging. I'll show it briefly. First, they tried only for 4 weeks, and they already seen significant improvement in positive and negative symptoms of schizophrenia. And now they extended it to 6 months, and it's still very effective. And believe me, this data are very impressive for psychiatrists to see. So they have very long-term effect, but what is most important, clinical safety. They have no side effects. They have just haloperidol without side effects. So what does it mean? It will be used in many other drugs. The haloperidol could be used in many psychiatric disorders, but they have very terrible extrapyramidal, metabolic, and so on side effects, and that's why haloperidol is not used in left and right. So it's only used in hard cases of schizophrenia. But the ulatorone now considered to be very interesting drug, very perspective. What about ralmitarone? That's a Roche compound with some number, was this TAR1 partial agonist. They just recruiting and performing clinical trials right now, no data published yet, so we don't know about that. So why suddenly TAR1 agonists become effective in schizophrenia? And again, making long story short, and I'm sorry for this very complicated slide, just look in the middle of slide. TAR1, as we believe, heterodimerizes with D2, so it's physically interacts with D2 dopamine receptor. So far, all psychiatric drugs, all antipsychotic drugs that are used in clinic, antipsychotic drugs, they all blocking D2 dopamine receptor. So that D2 receptor is a major target, as you see here. So now, instead of blocking D2 receptor, we have a partner molecule which is sitting next to that, which married D2. And by activating one, we changing property of another. And now this second receptor starting to signal different way. So it's changing signaling, blocking barrestin-mediated signaling, increasing G protein. That's some new concept. So what now is bias considered in pharmacology, bias ligand. So the idea is you can find a ligand, some drug, which activate different proportions of your signaling. So one side or another side. So now it's clear that when you activate GPCR, you have different waves of signaling. So you have not one signaling pathway, but several of them. Maybe we have considered here two, but maybe several. And Bob Lefkowitz actually and Mark Caron created this concept that to find biased ligands, which work only from one particular wave of signaling. So now here we have very interesting way of biasing receptors. So through partner. So you're not acting directly, not blocking dopamine receptor. You're working through one receptor and pushing signaling of second receptor. So dimerization induced biasing. That's why it's fine-tuning. So mechanism fine-tuning. You're not just hammering like haloperidol. Haloperidol just blocks the D2 receptor. That's why so many side effects. Here you just fine-tune it. So you just get the right way of affecting signaling. That's our explanation why it's working. Second explanation, it can be TAR1 is found in the prefrontal cortex and its modulates glutamate function. We published it several years ago. So, and it looks like it's NMDA receptor or AMPA receptor ratio is decreased, and you can modulate NMDA dependent currents are reduced in TAR1 knockout mice, for example, mice lacking TAR1. Even composition of receptors of change and phosphorylation of receptors is changed NMDA receptor glutamate. So that's second explanation. Third explanation, TAR1 can modulate serotonin function. And serotonin is also being involved in antipsychotic action and in schizophrenia. And there are several lines of evidence. I will not stop you here. You can see that TAR1 can modulate serotonin function. So basically, I was invited by Sunovian. I'm consulting them. I was invited to make a video to show how it's working. Why TAR1 is important target for psychopharmacology. And you will see it now. What Sunovian put together with Sunovian put in this video.
Рауля, у нас нет звука.
Нет звука? У меня есть.
Видео нету.
Нету, да?
Нет,
у меня есть. Я не знаю, как сделать.
Можете озвучить сами.
Okay. I'll continue. So, да. TAR1 is widely distributed through the brain, including in dopamine regions, such as ventral tegmental areas, substantia nigra, dorsal rachia, amygdala, prefrontal cortex, hypothalamus. Areas are important for pathophysiology of various psychiatric disorders. initial studies provided evidence for both intracellular and membrane localization of TAR1. They located straight postsynaptic. Functionally, TAR1 was implicated in modulation dopaminergic, serotonergic, glutamatergic system. That was shown complex crosstalk between TAR1 and dopamine system. For example, TAR1 activation reduces dopamine neuron firing. TAR1 inhibition enhances it. Additionally, TAR1 activation can inhibit serotonin firing, modulate glutamatergic synapses. TAR1's ability to modulate this system is important for mood, psychosis, reward processing cognition. That its potential as a therapeutic for several neuropsychiatric disorders, such as schizophrenia. Future research should characterize viable therapeutic targets to better understanding and bring better understanding of pathological disorders, psychiatric disorders, to this neurotransmitter. It was difficult, by the way. Okay. So they put the 13 citations here at the end, and I'm proud to say that 7 of them are our papers. How to make it further. Okay, so I'm sorry you didn't hear the sound, but I had it here. So what about other TARs? So TAR1 is now proven, validated target. Okay, so it's moving forward, and we're very happy about that. But what about other targets, other TARs? So the next best characterized receptor is TAR5. It was initially cloned from the skeletal muscle and brain, and it's considered to be putative neurotransmitter receptors. But later in 2007, 2006, very interesting paper appeared from Nobel Prize winner Laura Bach, who received Nobel Prize for discovery of olfactory receptors. She found that it's expressed in olfactory epithelium, so in our nose, and all of them except TAR1. So from TAR2 to 9, all these genes they found in olfactory epithelium, and they considered it like olfactory receptors responsible for innate behaviors. So, but what I really didn't like in this paper, they took whole brain, homogenized it, and said, no, it's not expressed in the brain. They are only olfactory receptors. And what they believed, that was not correct way of doing science. I'm sorry, because receptors can be expressed in a small number of cells. That's it. And then if you homogenize whole brain, you will never get it. What type of socially relevant odors are detected by TARs? It's decomposed tissue, rotten tissue. As I mentioned, it's decarboxylation. So when tissue proteins were decomposing by bacterial decarboxylases, you get trace amines. It's sexually relevant odors, pheromones, predator odors, like how mouse recognize cat or antelope tiger, for example. So there's a very interesting logic behind that. So predators eat meat. In meat, you have a lot of phenylalanine. Phenylalanine becomes phenylethylamine, and beta-phenylthylamine is the smell of beta-phenylthylamine. That's how mouse recognize cat, for example. So it's very interesting, simple logic here. So interestingly, the whole story of trace amines can be related to this old story perfume. Remember, many of you probably have seen this movie or read it, you know, about this perfumer who were collecting bodies of young girls and making perfume out of that. Isn't it interesting? Like it was like 30 years ago, way before these stars were discovered. If you remember in this movie, this baby, when he was born, his mother was selling fish, and she left him in in some played with a rotten fish for one day. He was the first day of his life he spent in rotten fish. It has specific order. For example, it's trimethylamine, and it was known, it was found that trimethylamine is the best agonist of TAR5, for example. This is a rotten fish odor. It's just amazing. I'm just amazed how Patrick Ziskin, I think his name, was predicting that TAR story, like rotten fish odor. That's the best agonist of TAR5. And antagonist, interestingly, has woody odor. Amber woody odor. The best antagonist is timberol, which blocks. That's another interesting thing. You know, some pleasant odor is antagonist, a very unpleasant odor is agonist of TAR5. And we have TAR5 receptor mice. Mice lacking TAR5, we created interesting one important point here. We can we inserted, not we, but colleagues who created them, inserted Lagzik neocassette. So it's not only knockout, so it's also marker. We have markers. So by studying for this marker, we can see where it's located. And our colleagues actually working, Stefan Lieberles, who was working with the same mice, they detected it very nicely in olfactory epithelium. You can see here, and you see on the top here, just on the beginning of the olfactory bulb, expression of TAR5 is blue. So it was shown here another way to look at. So it's starting from olfactory epithelium, projecting to olfactory bulb, and stops at the level of glomeruli. That's it. That was prediction. So it's not going anywhere. So they said it's just olfactory function. But when we started to study TAR5 knockout mice, we see a lot of changes in behavior. So they behaved like less anxiety. They showed less depression-like behavior. And we also see alterations in serotonin level in the brain. So how it is, how it can be. It's olfactory, why it's changing behavior, why it's changing neurochemistry. It looks strange. So, but when we started to look for LASI very carefully, we realized that it's expressed not only on the surface of olfactory bulb as it should be, as you see it here, NA, but also as you see at here, it's expressed inside of olfactory bulb. In fact, we see some projections which supposed to go inside of the brain, and we saw it in the brain areas where the projections go. So normally, that's what is happening. So when you get information from olfactory bulb, from olfactory epithelium to olfactory bulb to glomeruli, from glomeruli there are neurons which project in different brain areas, and all these brain areas are highlighted now, like hippocampus, amygdala, piriform cortex, and so on. So we detected it in limbic brain areas where projections go. And now we have even better pictures how to see it. You can see it here, how it projects. We detected even these projections. It's unpublished here. So very nicely going inside of the brain. So we now have some emotional brain areas. They are the most ancient brain areas involved in emotions. So now we believe that TARs are not only olfactory receptors. So there is new neurotransmitter system going from olfaction into emotions. Now the questions, to limbic brain area and emotions. Now, how individual TARs? So we detect with TAR5, is it important for other TARs? What are natural and synthetic ligands of TARs? And can we now target these TARs and get brand new treatment for disorders involving emotions? So depression, anxiety, and so on. Another important thing, we noted that TARs are expressed around ventricles inside, and in not only around ventricles, but the areas where neurogenesis occurs. As you know, there is adult neurogenesis happening in subventricular and in hippocampal brain areas, subgranular zone. So, and that's where we see TARs. And the question appears, why suddenly they appear where neurogenesis? And just think about that. These are sensors of rotten tissue. So when something happening that probably you getting trace amines rising, and maybe trace amines are sensing that and giving a signal that it's time to start producing new neurons. Just think about that. At the same time, we noted that number of dopamine neurons is increasing in TAR5 knockout mice. So we see increase in critical enzyme in synthesis of dopamine, tyrosine hydroxylase and substantia nigra in several brain areas. We've seen increase in dopamine level, increase in dopamine metabolite level, and even GDNF was a growth factor which is involved in dopamine neuron proliferation was increased. So is it possible that now, that's what again to show that, interestingly again, that you can stimulate neurogenesis by olfactory learning. So positive modulators, environmental stimuli, learning, exercise, estrogens, and antidepressants increase neurogenesis. And the negative is stress, glucocorticoids, aging, and so on. So and somewhere in the middle now we got trace amines which are sensing when something bad is happening. So now we directly tested neurogenesis by specific markers to show two specific double cortin, and we noted the increased number twice in TAR5 knockout mice, and PCNA, another marker of neurogenesis, was increased. So now, if someone will find the way to influence TAR5, you can potentially can change emotional behavior, change neurochemistry, change neurogenesis. So it's an interesting application. So now, is it important not only for TAR5 or other receptors as well? TAR2. Now we have the same thing with TAR2 knockout mice, and we found that TAR2 is expressed in limbic brain areas. The same way neurogenesis is increasing in TAR2 knockout mice. So it looks like it's common picture that several TARs can be influencing and limbic areas, emotional behaviors, and neurogenesis. And here we summarize that the TARs and the crossroad between innate olfaction, emotional behaviors, and neurogenesis. Okay, it is known that adult neurogenesis is mostly is really strong in fish, for example, in frogs. It's reducing to mammals. In mammals, it's much stronger in rats and mice in comparison to humans. And some people even debate that if humans have it. So normally, look at that. This is olfactory bulb in mice, in humans, and it's much smaller in humans. Olfactory bulb and neurogenesis zones are also smaller, but they still, I believe, and many other researchers believe, is important for humans. But who doesn't have it? Dolphin. Dolphin doesn't have any olfactory bulb and neurogenesis at all. And now I give you a few numbers. Zebrafish has 122, the highest neurogenesis and the highest number of TARs. Mice, rats, 14 to 17 receptors. Humans, 6, and dolphin, 0. Is it coincidence? I don't know. That's just the numbers. And we are working not only that part. So here are several papers showing that we're investigating a lot of other functions of trace amines, and in periphery, in the brain, and you can see the papers which are appearing right now from our lab. And the idea is we're trying to find new functions of this group of receptors. And we created all knockouts. We have our in vitro works, and I would like to thank a lot of people who were involved during all these years, for 20 years, and particularly people from my lab now. Recently, I hope I mentioned everybody here. And I would like to thank also a lot of companies that supported me for all these years, like Hoffman La Roche, Lundbeck, Arion, Biacat from Russia, and Axelena, particularly Axelena helping us a lot to develop this topic. I would like to thank everybody to listen to me. I hope I'm in time, Zhenya.
Well, almost. Thanks a lot. Now we will have time for 1-2 questions because we are a bit shift from our schedules. So if there are any questions, если есть вопросы, можно...
Да, задавайте вопросы, пожалуйста, если есть.
можно, yeah, I'll double it in Russian and English. So you can write the questions, or you can just put up the hand, and I will turn you on your microphone. Можно задавать вопросы на русском или на английском, как вам удобнее, если есть вопросы у кого-то.
Костя, ну давай, говори.
Да, слышно меня, Рауль?
Да.
Да, у меня такой вопрос, вот интересные очень данные по поводу нейрогенеза, да, вот эти их взаимосвязь с рецепторами TAR5, вот, и которые были показаны на нокаутных животных. Вот, насколько вот вы предполагаете, вообще, в принципе, возможно разработка каких-то лекарств, которые будут, там, как я понимаю, блокировать, наверное, да, рецепторы TAR5, и которые будут как бы помогать с нейрогенезом и помогать пациентам, людям, у которых это является такой, ну, существенной проблемой, например, там, в постишемическом каком-то состоянии, там, после инсульта и так далее.
Я считаю, что это очень перспективная тематика. На самом деле, чем хороши GPCR? 30-40% лекарственных средств на рынке, они тем или иным способом влияют на GPCR. Что это означает? Значит, все технологии разработаны. Достаточно легко и быстро провести скрининг, найти антагонисты. То есть это на самом деле, я боюсь, уже кто-нибудь делает. Мы затянули, конечно. Но это для компании вообще не проблема. Вот я слышал, вот, допустим, Хоффман и Рош, они за месяц 2 миллиона соединений прогоняют, если хотят. Ну, на скрининг, ну, как с ними бороться? Вот. Я считаю, что это крайне перспективно. Похоже, мы нащупали. Понимаете, было известно, что антидепрессанты, допустим, Прозак, вызывают повышение нейрогенеза. Это было известно давно. А теперь мы, может быть, нашли вот прямо более интересную точку.
Причина, почему это происходит, да?
У нас, похоже, мне кажется, гораздо логичнее. Это сенсоры гнили, это, ну, в общем, понятно. Что-то подгнивает в организме, надо добавить нейрончик. Ну, в общем, не знаю, мне кажется, что наша мишень куда более интересная, чем Прозак, например. Посмотрим, как это придет.
Спасибо большое.
Спасибо. Вроде у нас больше нет вопросов, так что спасибо большое, Рауль Радикович, за лекцию.